Groundbreaking discoveries often stir up more questions than answers, and nowhere is this more true than in the quest to understand how genetic variants in the RINT1 gene contribute to severe liver problems in infants. If you're curious about the molecular mechanics behind infantile liver dysfunction, as well as the controversies that still linger in the research community, you're in the right place. But here's where it gets especially intriguing. Recent studies reveal that the role of RINT1 might extend far beyond what we traditionally expected, linking it to pathways as diverse as autophagy, lipid metabolism, and even tumor suppression. 1. Arasaki and colleagues 2006 demonstrated that RINT1 orchestrates the precise positioning and integration of ZW10 into the syntaxin 18 molecular complex, which is critical for correct cellular trafficking between organelles. Imagine the crowded highways inside our cells. Without this meticulous regulation, cellular traffic jams can occur, compromising overall cellular health. 2. He et al. 2013 provided evidence for PTLNS3P-bound UVRAG in concert with ER tethers and the Becklin-1 complex as a key coordinator of retrograde Golgi ER trafficking and the transport of ATGAN9, an autophagy-related process. This showcases the interconnectedness of cellular recycling and secretory routes. 3. The importance of inter-organelle communication was highlighted by Xu and colleagues, 2018, who found RAB18 can physically anchor the endoplasmic reticulum, ER, to lipid droplets, LDs, via SNARE and NRZ complexes, thereby promoting lipid storage, an essential process for energy management within cells. 4. Davis et al. 2017 explored how secretory and autophagy pathways don't operate in isolation, but instead engage in complex crosstalk that ultimately shapes how autophagosomes are formed, a process crucial for removing damaged cell components. 5. Autophagy isn't just a mammalian affair. Chen et al. 2019 showed that it also depends on TIP20 in yeast, drawing remarkable parallels between simple eukaryotes like Saccharomyces cerevisiae and more complex life forms. 6. Here's a controversial revelation. According to Cousin and collaborators 2019, having mutations in both copies of RINT1 can actually cause infants to suffer from recurrent acute liver failure and unusual skeletal abnormalities, a dual phenotype that challenges the classical view of liver-only pathology in these patients. 7 through 8. Launay et al. 2023 and Quiros et al. 2024 advanced this debate by demonstrating that RINT1 deficiency doesn't stop at the liver. It can disrupt fat processing and lead to hereditary diseases like spastic paraplegia. This suggests a spectrum of diseases linked to the same genetic culprit, and it's something many clinicians still overlook. 9. In immune system research, Takeuchi et al. 2023 found that a hyperactive version of STAT6 amplifies allergic responses, highlighting how gain-of-function mutations can have far-reaching, sometimes detrimental consequences. 10. Jumper et al. 2021 presented the revolutionary AlphaFold algorithm, which now allows scientists to predict protein structures with unprecedented accuracy. This breakthrough technology is already transforming how we investigate proteins like RINT1. 11 to 14. Various Drosophila fruit fly studies by Schnorrer, Araki, Oka, and Kitazawa between 2010 and 2015 revealed genetic pathways regulating muscle formation, oxidative stress, and cell division, reminding us that key insights for human disease often come from studying the tiniest creatures. 15 to 18. From the geometry of protein helices, Barlow-Thornton, 1988, to the stabilizing influence of hydrophobic interactions, Tanford, 1962, Pace et al., 2011, Eisenberg et al., 1984. Understanding protein structure and stability is the bedrock of modern biology. But here's the part most people miss. Even the smallest disruption in these interactions, such as those resulting from rare genetic variants, can spiral into bigger problems affecting tissue function and health. 19 to 20, Peters et al., 2025, and Mann et al., 2023, highlighted an emerging consensus that defects in intracellular trafficking genes, namely NBAS, RINT1, and SCYL1, are frequent culprits of repeated acute liver failure in young children. Still, not every researcher agrees on the extent to which these pathways individually contribute, a point worth debating. 21. Grigorovicius et al., 2016, warned that inactivating RINT1 in the brain doesn't just cause cellular chaos. It kicks off genomic instability, ER stress, and blocks autophagy. Could this explain neurological symptoms in patients with liver disease? It's a hypothesis that deserves more attention. 22-23. Kabeya and Mizushima, 2000-2010, developed vital laboratory tools to study autophagy, focusing on LC3's role in mammalian cells, which have since become standards in cellular research globally. 24. Lin et al., 2007, controversially positioned RINT1 as a tumor suppressor, with roles in maintaining both the Golgi structure and the integrity of the centrosome, two structures critical for accurate cell division and overall survival. 25. Dombrowski et al., 2010, showed that genetic responses to ER stress, a common feature in liver dysfunction, can differ significantly between individuals, suggesting a genetic basis for variation even within similar clinical presentations. 26 to 27, reports by Saitsu et al., 2013, and Kim et al., 2016, offered glimpses into how mutations in autophagy genes, such as WDR45 and ATG5, can lead to neurodegeneration and developmental delays, parallels that could extend to RINT1 and related pathways. 28, G et al., 2023, found that a previously unknown NBAS mutation in an infant with fever-triggered liver failure directly sabotages this gene's function, strengthening the case for targeted genetic testing in children with unexpected liver crises. 29 to 32, Rao et al., 2015, Ye et al., 2017, Yorimitsu et al., 2006, and Gonzalez-Rodriguez et al., 2014 unraveled the links among ER stress, fat buildup in the liver, steatosis, and decreased cellular self-cleaning, autophagy. Such findings keep alive the debate over what tips the scale from adaptation to cell death during pediatric liver failure. 33 through 38. A string of works, including Weinman et al., 2012, Song et al., 2022, Ugor et al., 2016, Morez-Montaigne, 2021, Musselman et al., 2013, and Rustin et al., 2004, emphasize the versatility of Drosophila as a model for dissecting human metabolic, hepatic, and genetic disorders, enabling